Welcome to OmicsBox, bioinformatics made easy.
So the goals of this webinar will be to learn how to perform a basic single-cell analysis pipeline, starting from raw reads down to biological insights, the key steps to work with multiple sample scenarios, and how to explore our data in order to gain those biological insights. So for this demonstration, I will be using a dataset of 10 samples of Arabidopsis thaliana roots from 7 different seedlings named replicate A to J. And 3 of those samples were grown in a high sucrose medium and 7 in a normal medium. And the dataset was, the single cell libraries were obtained with the dropset library construction technique. So I won't go into too much detail of each of the steps, but this would be the pipeline that we are going to follow. We will first start with the quantification in order to obtain the gene expression values for each of the cells with the star-solo algorithm. Then we will filter lowly expressed genes and cells of that count table. And later, we will do the preprocessing of that count table because it cannot be directly imputed into the clustering algorithm. It needs further steps like normalization, dimensionality reduction, data integration, etc. And this will be performed with a SEURAT package, as well as the clustering. The clustering tries to find groups of cells with similar expression patterns that they should correspond to similar cell types or states. And after that, we will start exploring our data with the cell type prediction with Singular, which is a reference-based algorithm that compares the data with an already annotated single cell dataset in order to predict the cell types in our dataset. And finally, we will start asking questions to our dataset with a differential expression analysis with a single cell adaptation of EdgeR. And we will ease the interpretation of those results with a functional enrichment analysis. And all these steps are included or implemented into OmicsBox, which is a really user-friendly software with a graphical interface. So there's no need for coding expertise to run any of those analyses. And moreover, it's cloud-based, meaning that you provide your input data, it's sent to the cloud, the algorithms are run there, and then you retrieve the results. So now let's jump into the demonstration with OmicsBox. So the first step will be to perform the single cell quantification. First, we need to choose if we have cell barcode technologies as 10x, dropSeq, etc., or full-length technologies like SmartSeq or Smarter. We have cell barcodes. And on the first wizard page, we have to specify our input data. We need to specify the reference genome and the reference structural annotation. We have to specify how our exons are named in the GFF or GTF. And finally, the overhang parameter is the length of the read-containing transcript sequence -1. This is a parameter required by star-solo, and you can obtain this number by asking your sequencing facility or running a FastQC previously, and you will obtain the length of the reads in the results. And then we specify the FastQs here. This is something important when we are dealing with multiple sample scenarios, is that we have to provide one sample at a time. This is because the cell barcodes can be repeated in different preparations. So if we put all the FastQs of all the samples here, we could be mixing cells coming from different samples, which is not desirable, of course. So one star-solo run per sample. Then we need to specify where in the read does the cell barcode start and the length, where does the UMI start and the length, And here we have different presets of different technologies. So the parameters are automatically updated, or you can provide your custom parameters. We will continue with DropSeq. These are some parameters to fine-tune the alignment. I will leave them by default. And some other options to fine-tune each of the steps of a star solo pipeline. These are really well explained in the OmicsBox user manual as well as in the star solo documentation. So I won't go into too much detail here. And finally, we will give a name to our sample and we will click on run. This usually takes a little bit more than 1 hour to compute, but I already done this for you. Here we have the results. So the main results, as we see here, is a count table. We have the cells, the number of features expressed for each cell or for each barcode, the total number of counts, the features, the number of cells in which the features are expressed, and the total number of counts. And moreover, we have a summary report here with different statistics in how the alignment and quantification went. For example, we have the total number of reads mapped to the genome, unique, multiple, the the mean reads per cell, and the mean UMIs per cell, and genes per cell, etc. And we have this interesting plot as well, in which we have the total number of UMIs detected per cell on the y-axis, and all the cell barcodes detected in our dataset in the x-axis. This is because with droplet technologies, you can encapsulate in the droplets maybe contaminant or ambient RNA, so you will obtain a really low number of UMIs associated to that barcode. So we would expect that this number is decreasing until it reaches a point in which we see a sharp drop, and we have some barcodes with a really low number of UMIs, and we could expect that those barcodes are not truly cells, but rather they are background noise. And in green, we have the number of cells that have been kept in the count table. And we should expect that this threshold lays like right after the beginning of the sharp drop or close to the beginning of the sharp drop. So in this case, I would say it's correct. So as we see here, we have one count table per each of the samples. But in order to continue with the analysis, we need all the samples into the same count table. So I will use the merge single-cell counts for that purpose. Here I will browse for all the count tables. And here I can specify the experimental conditions just by clicking and typing. I can specify the conditions for each of my samples. And I can add as many factors as I want. And I will click on run. And here I have my merge count table containing all the cells for all the samples. And in order to avoid that the same cell barcodes are in the different samples, a prefix is added to each of the samples with the sample name. So we are sure that we have unique cell barcodes here. So now we can start taking a look at how our count table looks like. We can plot the total counts distribution for each of the samples. So here, the violin plot, the violin shape tell us the density of cells in that area of the plot. And the box plot gives us more statistical values of that distribution. And we see here that we have some cells that have higher number of counts in comparison with the rest. So that could be multiple cells called multiplets or doublets that have been sequenced together. So we would rather like to remove them before continuing with analysis. And it's really interesting to filter cells with a really low number of counts. But if we see here in the minimum number, like almost all our samples have a minimum number of counts of 500. This is because StarSolo already filters cell barcodes with a really low number of counts. So I don't see the need of specifying a lower bound here. And another interesting thing to see if the percentage of genes associated to mitochondrial or chloroplast genes in comparison with the total number of genes. This is because if we see a really high percentage of mitochondrial chloroplast gene expressed, that could mean that the cell has been broken, so only the mitochondrial and the chloroplast mRNA encapsulated in those organelles remain. So we don't really want to analyze those cell types because they have been broken and we have lost most of our RNA. So I would specify a cutoff or 10% or 20% in order to remove all these cells with a really high percentage of those genes in my sample. So in order to remove all these cells, I will use the filtering tool. So here, this is a really important step as well. This will ease the computational cost of following tools as well as to reduce noise, is to filter features that are not expressed in a minimum number of cells, 100 in this case. Here I can specify a minimum number of counts of 500, which is not filtering in my case, and a maximum number of counts of 20,000. And the minimum features, I will leave them by default, which is the minimum and the maximum in my dataset. And I will filter by the percentage of mitochondrial genes and chloroplast. I have to specify a file with the IDs of those features in order to run this. So I click on run again. And this, depending on the dataset, takes 15 and 20 minutes. I have my results, filter results here already. So if I plot the chart again, we can see that the cutoff has been correctly established. Okay, so now we will go to the clustering, but as I mentioned, we need to pre-process our data first. So this pre-processing is already included into the clustering run. So we can choose different algorithms to log normalize, specify the number of high variable features. We can correct for the expression of the mitochondrial and chloroplastic genes here as well. And this is another key point when analyzing multiple sample datasets, is that we need to integrate those samples or those counts. These algorithms are meant to remove the noise or the difference or the variance that arises because there are different samples and not really because of biological variance. Thanks to this, we will see later that the clusters will be composed of cells coming from the different samples. And this is based on the assumption that even in different conditions, the cell types that we can expect in the different samples are the same, but maybe they have differences in between the expression or in the abundance of each of the cell types. So we will integrate by the medium factor. And here we have different algorithms to perform the integration as well as other parameters. Finally, some other parameters to fine-tune the clustering and the visualization. We will click on Run. And this again will depend on the dataset, but it can take from 30 to 40 minutes. And here I have the results. So the main result is the count table again, but with a column with the assigned clusters and a UMAP/TSNE visualization, because we can choose if plotting the UMAP or the TSNE here. And by default, they are colored by the clusters obtained by the clustering algorithm, but we can choose coloring it by the factors that we specify and the samples. So as we can already see here, all the areas on the UMAP contain cells from all the samples, which is desirable. That is what we expected. And let me change this. I like them smaller. And maybe first thing to notice is that we have areas of the UMAP that we have more cells and that were grown in high sucrose medium in comparison with normal ones. So this visualization is already telling us some information. But in order to continue analyzing this data, we will go for the cell type prediction with a singular implementation. Here we specify our reference dataset. There are many databases online in which we can download datasets. And since they can contain many different types of cell metadata, we have to specify here which metadata in our reference contains the actual cell types annotation. And then this is very important as well, is that the genes on the reference and our dataset have to match. So we can specify in our dataset if we want to use the IDs or the names. In this case, they were the same. And if the reference contains many feature metadata, we can choose here as well the IDs of the reference. And if we are not sure which IDs the reference has, we can load it into OmicsBox and take a look. And some other fine-tuning algorithms for singular that are as well explained in the OmicsBox user manual. So I will click on Run, and it takes more or less 1 hour, depending on the size of both the reference and the dataset. And what we will obtain is an updated UMAP or TSNE visualization with the cell types predicted by singular, as well as some statistics that help us assess how well perform our classification. But I would go into this now for the sake of brevity. So here we can find the predicted cell types by singular. And singular, if the text cell labels that are a little bit ambiguous are unreliable, and it names them prune. And we see here that the prune labels are concentrated in the middle of the plot. And if we change to the Seurat coloring, we see that they are all belong to the same cluster. So that means that these cells maybe correspond to a cell type that was not present on the dataset, or maybe there are some spurious or unambiguous or rare cells that we should not trust. So as I still don't know, what I will do is start my own annotation. So I will select these cells and I will add it to my group name annotation undetermined. We will need to further investigate this cluster in order to try to determine what it is. Now let's further investigate our data. Okay. Here I have some cells that have been predicted to be in the G2M phase or the S phase of the cell cycle. So it is known that in the root, there's a cell type named meristem that is actively dividing. So these cells, they may actually belong to that cell type. So here I downloaded a list of marker genes. There are a total of more than 1,000 marker genes. I copy paste them and I can plot, well, I can color the cells by the expression of those genes. Let's wait a little bit. Yeah, here we have. So we can choose to plot the average, the sum, the minimum, or the maximum values of all those genes. So we effectively see that these marker genes are more expressed here in this area of the plot. So I think that now I will select those cells that moreover they are in the same cluster, in the cluster 4. So instead of with the lasso, I will select the cells in the cluster 4 and I will add them to my custom group. Merge them. And of course, I can see my annotation so far here. I have annotated these 2 clusters. So now we see the contrary example here, because here we have one cluster that is split into 2 different cell type predictions. And here we see 3 different main clusters that are predicted to be the same cell type, that is root endodermis. So why do we have the same cell type but different clusters? So we can try to see which genes have driven this separation into the clusters with the differential expression analysis. So first, we will filter lowly expressed genes. And then we have a simple design. We have biological replicates, samples. And now I will specify as blocking factor the medium, because I don't want this difference to interfere with this analysis now. I'm only interested in the different clusters. And the clusters I'm interested were the 10, 13, and 16. And I want to test each of them against the rest. So like this, it will take cluster 10 and it will test against this other 2, cluster 13, and it will test against this other 2. If I uncheck this, it will try to test all these clusters against all these clusters. so they cannot be repeated. But testing the contrast separately, this will remove the itself cluster from the reference. Another more options to choose for the statistical test. So I will click on run, and it usually takes 15, 20 minutes as well. And here I have the results. So what I would obtain is a table with all the contrast specified, with the statisticals computed by Edgar and attack if the gene has been found up or down regulated. And since I have a long list of up and down regulated genes, in order to ease that interpretation, I can perform a functional enrichment analysis to see if those up genes are more enriched in particular biological functions. So let's perform a feature set test for the up-regulated genes of cluster 10. Cluster 13. And cluster 16. So the Fisher exact test is really quick. And we see here for the cluster 10, we see a lot of enriched functions that have to do with the Golgi membrane and the transport of components. And this is because inside the root endodermis, we have different cell types. Well, with a little bit of biological context, the root endodermis, its main function is to regulate the flow of compounds in the membrane, in the epidermis of the root, and it has to build a membrane named Casparian strip. So in the bibliography, it has been found that there's an early stage of this cell type that has to still develop this membrane. So it is actively building compounds, and so we could expect that these functions are really enriched in this cluster, in this subcell type. So we can use the same tool as before, and we can select, the Seurat cluster 10 and assign it to early root endodermis. Okay. Then the results for cluster 16, we already see the Casparian strip here, and different functions that have to do with cells that already have this membrane and have to further suberanize that that part of the root. So as we are already performed here, we could select the cluster 16 and assign it to suberanized root endodermis. And finally, for the cluster 13, here we have a lot of functions that have to do with the response to stress. And in fact, if I color the UMAP back for the medium, we see that this cluster here has more cells that were grown in sucrose medium. So this could mean that even after data integration, these cells are so different in between the different conditions that they were not able to fully integrate. So these cells are enriched in functions that have to do with stress, meaning that maybe since this cell type is its main function is to regulate the enter of substances through this membrane, it makes sense that this cell type was super stressed for the presence of a sugar, of a compound in the medium in comparison with the normal ones. So we could continue this and this, taking a look into the clusters and the cell types and performing differential expression analysis in order to refine our annotation. But at the end, we were able to compare the difference in between, but we wanted to compare the differences in between the two conditions. And I will show you how to easily perform this. We will go now for the differential expression analysis. Okay. And now I will go for a multifactorial design. The biological replicates are the same. And now I would like to test for each of the cell types identified in my dataset. I would like to test the sucrose versus normal cells. And if I click on run, it will give me a results like this one. In which for each of the cell types, I compared the cells belonging to sucrose conditions to the normal ones. And I can further investigate this list as I've done with the functional enrichment analysis, or maybe taking a look at in the bibliography of those cells, painting heat maps, etc. But for the sake of time, I will end my demonstration here. Because I think that I already showed you how to conduct a single-cell analysis pipeline in a multiple sample scenario and the key steps to take into account. Then I show how to identify the cell types and the subcell types present in the dataset with the reference annotation and the differential expression analysis and with the biological knowledge. And yeah, for that, we used both the information given by the clustering and the cell type prediction. And finally, we were able to identify some differences in the gene expression of cells grown in high sucrose mediums. So this concludes my demonstration. If you would like an extended version of this analysis, you can go to the Biobank blog. I will put the link in the chat. And I would really like to thank you for listening to me, and I would like to thank the rest of the single-cell team at Biobank.
Thank you for watching. Learn more about OmicsBox with our other videos.